At some point, yes, but I think we're good.
Cool.
Hi, Adam. Can you make me a co-host so that I can share my screen?
Give it another try.
It worked. Okay.
Success.
All right, do you see what I see?
Yes.
Great. All right. There we go. All right. Thank you so much for your patience and thanks for having me. My name is Maya Hay and I'm beaming in from Chokchage, also known as Montreal. And I think it's particularly important to do land acknowledgements for virtual presentations when the cables, wires, and infrastructures that really go across unceded territories are really what enable me to share my thoughts with you today. And so I'm grateful that the conference organizers provide this virtual option, and I'm looking forward to hearing the other presenters at this panel. And in this presentation, I want to ask, what kinds of future organisms are we bringing into the world with technologies like synthetic biology and synthetic genomics, both of which seek to create new forms of life? And I'm part of an international team on a project called Future Organisms, and we're examining biological innovations in the UK, US, and Japan. So when I say we, I'm really speaking on behalf of a team of 6 social scientists. This work is funded by the Economic and Social Research Council and the National Science Foundation, and our research teams are based at 3 locations, the University of Edinburgh, Colorado State, and Keio University. And I've organized my talk into 4 parts, and I've really just taken the keywords from my title and rearranged them a little. And by the end of this talk, I'll hope that you have a better sense of why responsible research needs to consider the creatures and organisms who get enrolled in the technological innovations, and why the relationships we have with organisms will be an important part of the collective decision-making as genetic tools start to evolve and emerge. And I'll start by explaining why we chose this origami dragon as our logo. Firstly, it's because it's an organism that has resonances across all of the countries in the project, but also it's an organism that is arguably important in synthetic biology. For example, Ginkgo Bioworks, which is the world's largest synthetic biology company, went public last year, and they signed off their statement about going public with the line, "There will be dragons." And elsewhere, we see academic research labs like that of Andy Ellington, who I believe is also presenting at this conference, sign off their websites with, "Here there be dragons." In this instance, the phrase is less literal about dragons as creatures, and instead gestures to more figurative language, where research attempts to push the edges of the known world, even moving beyond it. In fact, the phrase, "Here be dragons," refers to a place on this map from 1504, where explorers didn't yet know what was beyond the charted world and the charted seas. It acknowledges unknown areas and serves as a reminder to be careful, as in full of care when proceeding into the unknown. And for us, this speaks to the sense of possibility at the edge of the unknown in both biology and genetic engineering. It also refers to the responsibility to face the unknown in considered, careful ways. So in both poetic and mundane ways then, this dragon acts as a symbol for the possibility and the responsibility of developing genetic engineering technologies, because it's a metaphor for our futures. And I want to now expound on some other metaphors that drive these innovations and influence how we imagine these technologies to be. At the start of my talk, I mentioned synthetic biology and synthetic genomics, and you may be wondering about what differentiates the two. And that's partly to do with scale. And for now, I want to highlight the fact that talk of writing seems to be the central to synthetic genomics, while talk of engineering is more prominent in synthetic biology. And there's lots more to say about this that I don't have time to go into right now, but I mention it because we often take the power of language for granted. Consider how DNA is thought to spell out the so-called book of life. And in this metaphor, each genome is a book and genes are discrete words within it. And the Human Genome Project was an attempt to sequence or read that book in full. And like any early reader, once you learn how to read, you learn how to write. So writing follows as a logical next step in our learning about genetics. And even the visual metaphors about writing and rewriting and erasing become prominent, even in logos such as the one for this conference. And here's another example where genetic code and computer code become one. The A's, C's, T's, and G's can be coded into zeros and ones and uploaded into living systems. And this gives rise to the idea that programmable cells beget programmable organisms, which leads us to this sticky question of what should be written. As Chris Voigt, a notable synthetic biologist, asked in 2017, now that we have the ability to synthesize anything, what do we build? What do we design with that capacity? And although I don't think it's quite the case that we have the ability to synthesize anything, this does raise the question of who chooses what to build and what to design? And also, what does it mean to design at the genome scale? Many genetic engineering projects are not really concerned with building whole organisms per se. They're just trying to build long stretches of genetic material. And particularly when it comes to organisms with big genomes like that of the human, projects are more likely to be at the chromosome scale. So the scale of intervention is important to us because each scale carries with it a different value. And here's what I mean. The scientific value of a gene or a cell might be one thing, but the social value of whatever organism carrying that gene may be very different. In other words, I have a different relationship to chromosomes than I do organisms. We relate differently to corn than we do a genetic piece of corn. And the same goes for cows, for rice, for pigs, etc. And that's because organisms come with established attachments to social worlds, both inside the lab and outside of it. And they come with rich histories, cultural significance, and an array of social practices. Just look at the way rice is cooked all around the world. And this means that introducing a new organism to the world is different than introducing a new genetic circuit or chromosome. So whereas synthetic biology tends to focus on discrete modular parts, synthetic genomics attempts to build entire new genomes or to learn how living systems work. And as the technical capacity to write genetic material increases, we want to understand the shift from parts-based engineering towards designing whole genomes and whole organisms. And this shift was predicted by systems biologist Bernhard Paulsen when he said, we will move from talking about genetic engineering of single genes to what may become known as genome engineering, where the whole organism is the context of the design. And that's really where our project is located at, where the whole organism is the context of design. And to date, a handful of projects have synthesized whole genomes, from small mycoplasma to larger bacteria to the yeast Saccharomyces cerevisiae, which is perhaps better known as brewer's yeast or baker's yeast due to brewing and baking capacities or applications. And the yeast genome is about an order of magnitude larger than anything else that has been synthesized before. And building on this, in 2016, a group of US scientists proposed the synthesis of entire human genome, although that project has scaled down its ambitions since then. Now I'll take a moment now to zoom in on the synthetic yeast project, commonly known as SC 2.0, because members of our team have been involved with them since 2015. And what was different about this project, they noticed, was that rather than attempting to engineer a discrete genetic part with a particular function, it was an attempt to engineer at the whole genome scale of the yeast. And there were lots of discussions among the scientists and how they were keen to preserve the yeastiness of the yeast, even in its synthetic form. And notably, SE 2.0 required collaborations with organism-specific experts. These were scientists who really understood yeast genetics, not just any kind of genetics. And the idea behind SC 2.0 is that yeast is a model system for understanding all eukaryotic cells. And much of this desire is to eventually understand human cells. But we see some other approaches to yeast as well. such as when synthetic yeast researchers approach yeast as though there are computer programs, talking about kilobases and megabases as though they are kilobytes and megabytes of unsorted data. And we also found that they work with yeast in ways that look more like interacting with a coworker, where they delegate work to it, they need to keep it thriving, it needs to be convinced to do its job, and so on. And that was how we got to talking with scientists about the nature of the organism and its idiosyncrasies. For example, one scientist said that what is distinctive about an organism is that it has its own agenda, which is to sustain itself. And because of this, an organism never just does one task. It always exceeds what we might want it to do or what we might engineer it to do. And contrast this to the idea of organism agnosticism, which is a common idea with synthetic biologists who see a cell as a stripped down frame to load mechanical parts onto. And this is actually a metaphor that was brought in from mechanical engineering. And so with a chassis, which organism we use doesn't matter as long as it loads the construct we want it to hold. And while we certainly see some of the chassis metaphor in genome scale projects, we also see more of the organism being featured in projects of today. And in fact, we just published a paper that helps us make sense of how the organism is featured in genetic engineering. And we've developed a tagging system that will likely expand, and this will help us categorize how each project uses an organism. And we're hoping to find patterns across our 3 countries of US, UK, and Japan. So as science becomes able to write entire genomes, we're seeing a turn to the organism. And arguably, if we look at the Green Revolution and its aftermath, public outcries against GMOs suggest that consumers were always interested in the whole organism, not necessarily its parts. But scientists are now interested in as well. And this relationship between scientist and organism, between researcher and researchee, is something that we really want to analyze, especially as technological innovations increasingly come into our lives and livelihoods. Now, the working relationships we have with organisms is important to us because we have a long history of using each other. We've used plant and animal species to our benefits, just as they've used us to survive, adapt, and evolve. And we have been evolving together. Of course, what is both interesting and concerning with genetic engineering is that of possibility. Because if we have the possibility to design anything, then you can design something that's never existed before, something that never came about through evolution. And so as the famous last sentence of The Origin of Species puts it, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. And genetic engineering arguably picks up the "and are being" bit of that sentence. And the range of possibility is illustrated by this picture of speculative creatures, which was on the cover of the New Yorker last year. And here the artist says, I wanted the drawings to look like animals that could have evolved, but didn't for some reason, to remind people that things might have turned out differently. And we think that this speculative work is something worthwhile. Genetic engineering has the potential to develop future organisms that we've never seen before, such as those depicted by artist Luigi Serafini in an illustrated encyclopedia. It's an imaginary world with imaginary creatures, and it's this imagination that helps us go beyond what is doable and profitable to really test the integrity of our value systems. And I'll give you a tangible example of what I mean. A little over 20 years ago, a group from Symbiotica used stem cells of pigs to grow them into wings, under the premise that winged creatures have been a part of myths throughout history and across cultures. And importantly, the shape of the wings would often serve as a moral indicator of good or evil. Think of how angels would have the shape of bird wings or bat wings on demons. And in using pig cells, this project asked, how are we going to treat animals with human DNA? How will we treat humans with animal parts? And what will happen when these technologies will be used for purposes other than strictly saving lives? Now, in light of recent xenotransplantations from pigs to human, and I'm thinking specifically here about kidneys and hearts, these questions from 20 years ago are salient now more than ever. And thinking beyond the realm of the possible, often through art, helps us get through the sticky questions. And this is why we frequently collaborate with artists and designers who help us see the interconnection between the scientific, sociological, and cultural forces at play when innovations emerge. In fact, our team has a track record of working across disciplines and working with scientists to really open up questions about what is even possible, or what does it mean to apply engineering principles to living systems. For instance, this edited volume, Synthetic Aesthetics, was a major collaboration with Drew Endy at Stanford, artist Daisy Ginsberg, and Christina Agapakis, who is now with Ginkgo Bioworks. And our work with scientists and artists comes from asking questions such as, how does science work? What makes it good? Good for whom? Good under what circumstances? Who gets to decide? And at what stage do they get to participate in that decision? And these are all questions from a field called Science and Technology Studies, which is an interdisciplinary field that examines the intersection of social and technical developments, when there's a particular focus usually on historical contexts, cultural contexts, and political contexts. And one aspect of STS engages with the question of what does responsible research mean and what does it look like? And we want to use organisms to help open up the domain of responsible research and innovation, or RRI, which has come basically to become a standard way of organizing conversations about science and society. RRI is in many ways an outgrowth of ELSI, which stands for Ethical, Legal, and Social Implications, originally popularized by the National Science Foundation for the Human Genome Project. And one popular definition of RRI comes from Jack Stilgoe, who called it taking care of the future through collective stewardship of science and innovation in the present. And we've recently come to think about what that collective means in practice in terms of making space for not only diverse perspectives, but perspectives beyond the human. Because at the end of the day, human well-being depends on the well-being of other creatures. So conversations about interests and risk and benefits and goals and outcomes of research are really about being a good human in this more than human world. And so with that, I'll leave you with our coordinates. This is our Twitter handle. You can reach me by email. And the slide deck that I just whizzed through is also available on the link here. And in fact, there's a video that I skipped in the interest of time from our fearless leader, Jane Calvert, that I've linked in there as one of the skipped slides. So you're welcome to that. And I'll stop sharing my screen. Thank you so much.
So any questions from the audience now? Got one coming up.
So that was great, thank you. When you think about new organisms, it seems to me there's an ethics component because they're by definition new, or their definition a creation, but they're life, right? So how do you see in this biosphere of life, or sort of equivalent citizens on planet Earth, how do you see new, truly new synthetic life in that range in ethics?
Yes, the ethics question. And the simple answer to it is ethics is always plural. There is no one ethic that will be applicable to all circumstances of all places at all times, right? Ethics is continually a line that we draw in the sand, and it is always moving, and it needs to keep moving. And so rather than answering that question of, well, this is going to be ethically good and this is going to be ethically bad, I would say that this is why social science and humanities perspectives perhaps ought to be involved earlier on when designing these types of experiments and projects, so that it doesn't become an afterthought.
That was terrific, and I needed time to catch my breath afterwards, so thank you very much. What I'm wondering is that, of course, it's a slippery slope, as they say. We make one change at a time in humans or a cow or a plant, whatever. And at some point, we're at a new organism, well, species, and then at a new dragon, if you will. How do we know where we are on the slope?
That is the million-dollar question, Perry. And I think that's why I don't want to speak on behalf of any government organization, because I certainly don't work for the government. But I would venture to say that's why the whole population question that was asked earlier, I'm assuming that's why that there's been no development on that front, precisely because this question of how do we even make that decision is an extremely difficult one. And so when we talk to scientists and when we talk to regulators, the answer that we've gotten so far is we'll know it when we see it, which is the least satisfying answer on the planet. But nevertheless, that is where we are at. And so part of the work that we're trying to do on this project is to talk with people who are at the forefront of this type of research to be able to see what is the tech, where is the technology, where is the desire, so that policy isn't lagging behind 3 years, like too late, right? And in a way, that's kind of where RRI is right now. And so is ELSI. It's a bit of an afterthought and it's kind of lagging behind. And so what we're trying to do is to, get everything up to speed and be able to think ahead, so that there isn't this catch-up process that we seem to find ourselves into every single time, so that we can just move on from the we'll know it when we see it type of response, right? I know that's a non-answer, but it's the best that I've got.
Thank you.
So as a kind of piggyback off of that, have you seen at all what broad public thinks of when they'll know it when they see it? What is their definition at all?
Unfortunately, I do not do the public engagement work, and that's not to say that it isn't important. It is crucial. And if people were not able to listen in on Riley Tidingfung's presentation, please find her papers, because it is incredibly important how we go about and even imagine what public engagement even looks like, right? And even in her presentation, she was talking about how do you build those relations early and often so that there can be participation and agency from the start. So rather than ask the question of, well, what does the public think? I would say, how can you involve the public so that you can answer that question yourself early on in your own designs? And I don't mean you, Adam, I don't mean to pick on you, but as a conversational point, right? To really involve multiple perspectives from the start. That's, I think, going to be crucial.
All right, thank you, Maya. Looks like that's all the questions here. And I don't see any in the other chats. So with that, I'll introduce our next speaker, Julia Davideva, from right here in UMN. You should be.
This is good. Okay, yeah. Hi, everyone. So, thank you so much for inviting me here. And for those who don't know me, I'm Julia Davidova, and I work here at the University of Minnesota Surgery Department. But before that, 22 years ago, I joined the Gene Therapy Center at the University of Alabama at Birmingham, and that was a very fun place, especially at that time. And that was exactly the place where I fell in love with gene therapy and adenovirus. And adenovirus caused the common cold in normal life. Speaking of which, you can hear my accent and I can't hide it. I was born in Siberia, in the middle of nowhere, and I spent 30 years there. And that was a very cold place. We used to say we have 3 cold months, other 9 months are very cold, which is very true. And now I work with the cold virus, and I live in the coldest city in America. And I do love winters here in Minneapolis. So you can see I have a lot of cold here. So, but let's go ahead. So when we think virus, we think disease, and that's right. Viruses have reshaped and destroyed the whole civilization. That was cowpox virus. Let me go a little bit back. Yeah. So that was cowpox virus, which is responsible for the decline of both the Aztec Empire and the Inca population. Smallpox virus didn't spare the royalties. It killed the Egyptian pharaoh Ramses V in 1145 BC. And later, cowpox was the leading cause of death in 3 centuries in a row. So in the 18th, 19th, and 20th centuries, taking about 400,000 lives every year. On the other hand, the very similar virus, cowpox virus, saved millions and million lives when it was used as the world's first vaccine. And that was English doctor Edward Jenner who described the protective effect of cowpox against smallpox in his famous book with the long title. And Dr. Jenner is often referred as a father of immunology. But what I can say that this work, his work, perhaps saved more human lives than any other works. And a very close relative of cowpox virus is Vaccinia virus. And this virus was used to eradicate smallpox in a global vaccination campaign in the second half of the 20th century. By 1980, smallpox was eradicated, and now we really rely on this virus because I think that's what we can use for monkeypox. And that's the very updated news on this outbreak. So what we can see is that, and actually, I'm sorry, I can get used to, yes, okay. So, and of course I can't miss the latest pandemics. We all know that 4 adenovirus-based vaccines were used for COVID, including Oxford AstraZeneca and Johnson & Johnson vaccines, and all of them demonstrated a pretty good safety profile. So what means that viruses are dangerous, and yet humanity can benefit a lot if we can use them wisely. And I truly believe that viruses, yes, very my apologies, but I truly believe that viral-based gene therapy approaches and viruses particularly are the smartest pharmaceuticals on the current market because, first of all, they can multiply, they are alive. So what make them different from non-viral gene therapy approaches? They're alive, they can multiply, they can produce viral progenies, they can replicate, they can eliminate cells as a part of natural lytic cycle, so they can use the cells as a virus factories, and they're smart, so they can outsmart the host immune response. And finally, and what's most important, for million and million years, viruses have been delivering their genes into different type of cells, humans, plants, animals. So they're naturally made gene delivery devices. And so why don't we use them for cancer treatment? And eventually that's not the new idea. In the midst of the, in the middle of the 19th century, doctors have noticed that both influenza and the common cold were often associated with the tumor regression. And that was the time when we started using the viruses for human diseases. And then later on, in the 1950s, 1960s, that was a number of pretty much bizarre experiments when patients were treated with impure virus lysates. We didn't know how to purify these viruses at that time, so that's why most of the patients were treated with just infected body fluids, which were administered via all imaginable and non-imaginable routes. And that was exactly the case when in 1956, 30 women with cervical cancer received the treatment with wild-type adenovirus 5. Most of the viral particles were arrested by the immune system, but surprisingly, tumor regressed, and that was no severe toxicity. And most of these first clinical trials were conducted by Dr. Chester Southam, who teamed up with Alice Moore and conducted more than 150 clinical trials. And for sure, I think that he's the father of the oncolytic virus therapy. Even though now he's considered to be rather like a zealous oncologist, because at some point he stopped working with the animal models and started transplanting human tumor cells, HeLa cells, into human volunteers. And of course, this case was heavily criticized, but at that time we didn't have RB and all those regulations. And this story is very beautifully described in this book, The Immortal Life of Henriette Lacq. But now we live in a very different era. So that's the era of genetic modifications, when using the line from I'm a Legend movie, we can take something designed by nature and reprogram it to make it work for the body rather than against it. And it's pretty much what we are doing now. We are using the plot line of an apocalyptic movie to improve cancer treatment, and it works. There are different viruses which could be used for cancer treatment, and all of them could be genetically modified to make them safe and more efficient. There are different oncolytic viruses. So we have measles, we have retroviruses, we have lentiviruses, we have herpes simplex virus. That's a very interesting virus because the FDA has recently approved the herpes simplex virus for the immunotherapy for melanoma. We have Vaccinia virus. That's Vaccinia virus is the same virus which was used as a vaccine against smallpox. And we have adenovirus. And I'm here to advocate adenovirus because that's the viral vector we work in my lab. So, and indeed, adenovirus has a number of great characteristics. As I mentioned before, adenovirus in normal life causes the common cold. So it's not associated with any severe disease, it's safe. Adenovirus doesn't integrate into our chromosomes, it just hits and runs. Adenovirus can stimulate the robust immune response against cancer cells, which is great. We are so fascinated with the immunotherapies lately. And what's great for us is its cloning capacity. We can easily incorporate huge pieces of DNA into the adenovirus genome. The very first oncolytic adenovirus was designed in 1992, and I remember that time, and that was Dr. McCormick and a small pharmaceutical company called Onyx, which designed this virus, which was able to replicate specifically in P53 mutated cells. That's what we thought at that time. And you can imagine the excitement of the scientific community and the health providers when such a magic bullet was introduced to the market. Soon after that, they launched a series of clinical trials, but somehow these clinical trials showed somewhat disappointed results. The virus was not effective. It was safe, but it was not effective. And soon after that, and thanks probably also Jess Gelsinger case, this project was dropped by Onyx. A bit later, Shanghai Sunway Biotech company purchased the world rights to Onyx technology. The virus was renamed and then later approved in China for the treatment of brain cancer. So how come the first Antiviral virus didn't make the bigger splash in the field of cancer therapy? And in theory, even one viral particle, oh, finally, you can see, even one viral particle can destroy the whole tumor as long as it can efficiently replicate in there. However, it's never the case, even in vivo models. And in general, the experience from the first clinical trials with oncolytic adenovirus demonstrated very limited spread in solid tumors, and as a result, a very poor anti-tumor effect. So what can we do to overcome clinical limitations of oncolytic adenovirus? There are different approaches, and I can't cover all of them in the span of 15 minutes, so just I can probably introduce the most common and powerful ways we are using in our labs. First of all, now we know the reason why the first clinical trials were not effective. It's explained by the fact that most of the cancer cells are profoundly deficient for the adenovirus primary receptor, which is called Coxsackie adenovirus receptor. So what we can do, we can retarget our viruses to other receptors than CAR. So it could be integrins, and we can design the RGD modified vectors which bind to integrins, which are overexpressed on the surface of cancer cells. Or we can make a chimeric vector. We can replace the AD5 knob, which is responsible for binding, with the knob taken from another adenovirus serotype. So it would bind to CD46 and human desmoglein 2 receptors, and that's how it works. So here we can see that AD5-free vector showed one order higher transduction in the orthotopic pancreatic cancer model. And of course, we want our vectors to be specific, and so we can control adenovirus replication with the help of the tumor-specific promoter. And here we can see that COX-2 promoter controlled adenovirus showed no replication in normal pancreatic cells and a very robust replication in pancreatic ductal adenocarcinoma. And of course, I think that's a very powerful way to arm our vectors with the therapeutic genes, the therapeutic and imaging transgenes. So an adenovirus can be used as a vector to deliver a gene of interest to human cells. And there are different genes which could be incorporated. In our lab, we are using cytokines to initiate immunotherapies, and I believe that's a very promising approach. And the second vector used which was designed in our lab was designed to encode a very special dual function transgene called sodium iodide importer. So we could facilitate the imaging and radioiodine therapy for cancer patients. So what is the NIS? NIS is a normal protein. It's localized in our thyroid, and it's responsible for uptake of iodine. So, and the natural localization of NIS in thyroid has been utilized as a way to achieve radiotherapy of thyroid cancer. So people don't die of thyroid cancer for more than 50 years because we can do visualization of this tumor using iodine 123, and we can prescribe radioactive iodine 141 orally. So the idea was to deliver the sodium iodide importer gene with our adenovirus to other cells than thyroid. And that's how it works. That's a phase 1 clinical trial of NS-mediated gene therapy for men with locally recurrent prostate cancer. And this trial was conducted in Mayo Clinic by our collaborator, Dr. Morris. And here we can see the SPECT-CT image of iodine 123 uptake in the prostate after a single dose of the first generation NS expressing vector. So that was a first generation adenoviral vector, which was not selective and which was replication deficient. What is more important here, that this patient responded very well to the following radiotherapy with radioactive iodine 121. So, but before we go there, it's a long way before we go to the clinic, to the clinical trials. Of course, we have to study and evaluate our vectors in vitro, and then we have to evaluate their efficacy and safety in vivo models. And very often we are using patient-derived xenografts, which are being implanted into immunodeficient animals. And of course, we analyze the survival rate of our adenovirus-based therapeutics. Here we evaluated the survival when our adenovirus was combined with radioactive iodine therapy. And of course, we do research on the combination therapies when we combine our adenovirus-based vector with the standard approaches as chemotherapy or radiotherapy. And here, for example, we can see that the effect of interferon expressing vector when it was combined with 5-FU and radiation. Interferon is a very powerful immune cytokine. It can stimulate the immune response, plus it's a very we can use it as a way to sensitize radiotherapy and chemotherapy. So, and when we don't have the models, we create our own models, and we are trying to develop a gene editing pig model of pancreatic cancer, because we learned that pigs, as us humans, they develop cancer with age. So, and eventually we don't have any available pig cancer models available, and we really need that because adenovirus cannot replicate in murine tissues, unfortunately. So the pig model is the best model to study the true safety and efficacy of our adenovirus-based approaches, especially if they're administered systemically. And then we designed the toxicology studies, but before that you have to meet with the FDA, and we are lucky here because the FDA has approved our protocol to conduct the IND-enabling toxicology studies for Oncolytic adenovirus in pigs. And they also approved our way to manufacture the preclinical lot for the toxicology studies in my lab. And then we have to manufacture the virus, and that's the most expensive step so far, because it has to be manufactured by the third party. It has to be the FDA accredited facility, which is able to produce the GMP, good manufacturing practice level product, and it costs a lot. It takes a lot of time and costs a lot of money, and sometimes more than 1 million. So what I want to say here, that yes, we can design and produce all these exciting vectors. We can enhance their infectivity, we can make them safe, we can make them more effective, but it's not enough. The biggest overall challenge is how to get new viruses clinically tested. And here I'm talking about this enormous amount of work and expenses, mostly required to move your new viruses to the clinics. So, but I truly believe that nothing is impossible, and we have a hope. And especially now, I think that we live in a very exciting time, because the risk historically associated with virus therapy is lessening. And we do have increased enthusiasm among NIH regulators and biopharmaceutical companies, which is great. And I think that it happened because of the fact that 4 out of 4 vector vaccines for COVID are based on adenovirus. Once again, and then million people were injected with adenovirus-based vectors. So, and of course, we do have a hope to open our own phase 1 clinical trial, and we make everything to make it happen. And that will be a phase 1 clinical trial of adenovirus-based radiotherapy for patients with locally advanced pancreatic cancer. And here we will administer our sodium iodide importer expressing adenovirus, and then later we will visualize. We can also quantitate the NIS uptake using iodine 123, and then we will test the safety and efficacy of adenovirus-based radiotherapy. We will administer radioactive radioiodine 131. So with that being said, thank you so much for your attention, and thank you everyone who helped us to move our adenoviruses towards the clinic, and I'm ready for your questions. Thank you.
Hello, that was a pretty fantastic talk. How far do you think you are from, or do you think it is possible for using this adenovirus-based immunotherapy for more advanced cancer, or like more metastasized, rather than like, oh, I guess like in addition to treating the localized cancer at this point?
Yeah, that's a brilliant question, and we do discuss it a lot, and it's all about the way you administer your adenovirus. As long as your virus-based vector can work efficiently upon systemic administration, you can reach metastatic disease. And in case of adenovirus, it's really difficult to judge the efficacy to treat metastatic cancer using the murine models. And the reason is because human adenovirus is very species specific. It's human adenovirus, so it tends to replicate in human cells only. So it doesn't replicate in urine. So when you are using mice systemically, your virus will go directly to the human xenografts. And of course, we do see a beautiful therapeutic effect. We don't see any toxicity mostly, because once again, human adenovirus replicates only in human xenografts, in human tumors. So that's why we need the peak. But I truly believe that the systemic administration of virus is possible, and so that will be our next step. Once we run this phase 1 clinical trial, the next step will be the systemic administration with the hope to treat metastatic pancreatic cancer. Yeah.
Pretty cool, thank you.
Thanks.
So viral immediately has a negative connotation, as you've noted earlier. When sharing to the public, what is something you'd like people to take away from oncolytic viral therapies as opposed to viral by itself?
So, yeah, similar to the previous speaker, I'm not that much engaged with the public, which might be, yes, not that good. So, but I think that my impression is that the general public doesn't have any fear towards virus-based therapeutics, especially now. I don't see it. I think most hassles come, it's very strange, from the NH regulators or the FDA regulators. So, but that's my vision, that's what I think. But of course, I think, and especially now, during the last years, when we are so educated, I mean, we just, all of us, we are so much educated about the vaccines for COVID and how it works. I think that we are not going to have any problems in this direction. That's my perspectives.
I got a question. So,
Sure.
When you administer the oncolytic virus, there will be an immune response against the oncolytic virus. So, how much time do you have for this to spread into, amongst the cancer cells and cause lysis and release antigens that can be recognized and get you the anti-tumor effect that you're looking for? How much time do you have before the antiviral response kicks in and then shuts it down?
Yeah, that's a great question. So I'm trying to figure the way how I can answer it briefly. So first of all, it all depends on the route of administration. So you shouldn't be concerned about the immune response once you administer your virus locally, intratumorally. That's fine. So secondly, we had a number of clinical trials which showed no elevation in pre-existing antibodies after systemic administration of adenovirus. Thirdly, what we learn from the use of adenovirus-based vaccines, it's not that immunogenic as we thought. Still, that's a concern. So, and in reality, I think it's very important for us, and especially if we consider the intravenous administration of adenovirus to treat the systemic disease, to improve the efficacy of our adenovirus. So because it's always like a competition, what goes first? The oncolytic adenovirus, which reach the tumor and kills the cells, or the immune response, which will just simply eliminate the viral particles from the bloodstream. So that's why I think it's so much important to make our vectors more efficient rather than more safe. So because we are so much somehow focused on the safety of adenovirus-based therapeutics, while in my understanding it's vice versa. We have to be more focused on the efficacy of adenovirus. So adenovirus would be able to eliminate the cancer cell before its own elimination by the human immune response. That's a great question. Yeah, Daniel.
That was going to be my question.
Okay.
It's great talk and visionary talk with history. Yeah, and just one comment to you, question. So I think the public is ready for a virus therapy and investors ready for it, and there's big acceptance of it. And you show how we can destroy things using viruses. So from your perspectives, what are other targets for gene therapy? What actually we can repair by viruses?
So are you
Some genetic
talking about cancer diseases or monogenic diseases?
Some genetic diseases.
Oh, that's, yeah, we do have the specialists here in monogenic diseases, inherited diseases. I think that regardless the vector system you use for your studies, it would be AAV, it could be adenovirus, it could be Vaccinia virus or measles virus. I think that it's a very promising approach to help patients with both the cancer disease and inherited or monogenetic diseases. Again, as every target disease, speaking about the target disease, You know, I think, yes, I listed so many viruses. None of them is perfect. None of them. I think what we have to do, we have to think twice before we apply this virus for some specific target disease. We have to think twice, like pancreatic cancer, for example. I think the best way to reach it would be, yes, adenovirus, measles viruses. For AAV, that would be different target diseases. And of course, the doses and routes of administration. Everything needs to be taken into account.
And now, in the age of synthetic biology, we actually can create new viruses.
The new viruses. But eventually they're all new, yeah, because they're all genetically modified, but it could be even more. On the other hand, I would like to say that our ability to produce all these exciting new viruses, genetically modified viruses, greatly outshined our ability to test them clinically. So, and in reality, so the viruses which finally reach the phase 1 clinical trial are not new. They're like 7, 8, 9 years old. So we're so much ahead of it. But still, yes, that's always good to think about the new viruses. Yeah, sure. Thank you.
And so while we're, just before we go on to our keynote speaker, I'd like to thank the University of Minnesota Center for Genome Engineering for being a co-host on this meeting. They've been a huge supporter and a big part of why we were able to run this both virtually and in person. And so hopefully made that accessible to everyone. Now I'd also like to thank our remaining sponsors, who have also been a huge contribution to this. So with that, I want to introduce our keynote speaker, Sebatian Cocioba, who is from Binomica Labs, and I'll let him present.
Hi, friends. So my name's Sebastian. Full disclosure, I'm an amateur biologist. I hold no formal degree whatsoever. Did 3 years at Stony Brook University, then dropped out for family and financial reasons. But I still want to be a biologist. So regardless of what my vocation is, what work I'm doing, I still want to practice biology and explore the living world. So I'm going to give you a little bit of an introduction about some of the projects we're doing, me and my degreeless heathen friend, Sungwon Lim, in what was the 3rd bedroom of my mom's apartment, and now a windowless dungeon artist space, which is an interesting lab space. Okay, so without further ado, let's begin. So a bit of perspective. This is us, but from really far away. So this is taken from the Voyager 1 spacecraft, and it's the furthest picture of Earth ever taken. And for perspective, that's us. So that's everything. As Carl Sagan said, everything that anyone that's ever lived or will live will probably be there. So on this pale blue dot that I love so much, there's this weird green stuff that I'm going to talk about a lot that contributes to every breath you take. It contributes to your energy. It contributes to the entire ecosystem. And it works on a mechanism that I love so much called life. So here is a very eukaryocentric and very skewed tree of life. This is what they use in some like middle school textbooks to explain the diversity of life. On this planet, there are many things. I like the really green stuff over there, known as plants. So I love plants. I love plants so much. This is Romanesco broccoli. It has an actual fractal pattern building in there. So there's a mathematical equation being applied through just simple interactions, cell-to-cell interactions. And there was a recent paper that actually modeled that and replicated it in Arabidopsis, which is fascinating. Plants are also enormous. This is General Sherman, 83 meters tall. It moves water up and down the trunk of its tree each day using essentially nanotechnology. You need about like 40 kids to hug it in circumference. Enormous, enormous creatures. But my main love is this. So these are flowers. One day, if I ever grow up, I want to be a flower designer. That's my goal, a molecular florist of sorts. But when you're taught flowers in middle school, you see something like this, right? And it's very simple anatomical model, nice macro ovary so you can see all the parts. But plants are weird. Like, for example, Fritillaria melangensis is the second largest genome on the planet, upwards of 86 gigabases, polyploid, terrifying. The largest genome on the planet is held by Paris japonica, which is 156 gigabases diploid. Sequencing that, if anyone want to fund that, I'd be more than happy to do the legwork for it. On the other side, we have the twin orchids. It gets weirder. Like, for example, like what is that? That has nothing to do with that flower model that we saw earlier. It gets even weirder. Like, there are orchids that look like ducks and strange spirals. There's also structural patterns. Like, for example, on the right, we have the blue morpho butterfly. On the left, we have a begonia species that, when shining light at a particular angle, has the same structural color modality as the blue morpho butterfly. So all those tiny pits and structures that are built can be seen. So that blue is not a blue pigment, but a reflection of light that bounces off blue light specifically at that angle. But nature is not just something to awe at. We also have tools to kind of understand it. Like, here we have an example of, if you truncate a transcription factor just enough, you can shrink certain petals. You can get essentially different species of flowers using this type of small modifications. You can also do weird things if you screw up. Like, for example, here are particular transcription factors. When they're overexpressed, they start to shrink certain petals. This may not be aesthetically pleasing, but I love this. This is one more paintbrush to add to a palette. But here's something crazy. We also have other organisms that impact the morphology of plants. Like, for example, here's the Spindle Gall, and it's caused by mites. And the mite eggs, when they're laid, secrete hormones of auxins and cytokinins that change the cellular fate of the cells on the plant that they're touched to produce these wild structures. And inside that structure, you actually have mites growing, living, laying eggs, surviving. And this structure is nowhere in the host organism's genetic makeup. We don't have, like, oh, here's the patterning for this particular thing. This is hormones over time. Hormones over time. This is an oak leaf producing what looks like Cheerios, and it's like fuzzy and strange. Again, nowhere in the body plan of an oak are these things. It gets even weirder. What is that, right? Like, that's color and shape. There's textures to it, particular volatiles. This is from an oak gall wasp. The inside, when you cut it, isn't just hard tissue, it's substructures, it's complexity. There's so much nuance going on, and yet there's no genetic engineering here. This is secretion of hormones over time. But my story started with this. So this is a recreation of the very first thing I've seen under a microscope. My mom let me sacrifice one of her lucky bamboos, which isn't a bamboo, it's in the allium family. And we used some fountain pen ink and dyed it. Luckily for me, that was crystal violet, so it had some really nice binding. We washed it with some alcohol. I saw it under a kid's microscope, a Fisher-Price microscope, and that blew my mind, because that was the first time that I saw cells from the textbook to the real world. I literally saw all of the compartmentalizations. And that microscope under my bed, fast forward a couple of years, turned into this. So this is roughly what I have at home. This is the third bedroom of my mom's apartment that I turned into a fully functioning molecular biology lab, complete with biolistic gene guns, laminar flow hoods, PCR machines, you name it, everything to do biosafety level 1 in an intimate home space. So here's my lab bench on a good day, right? I love flowers tremendously. Petunia is one of my favorite models. My bread and butter is in tobacco, but Petunia is something I really want to strive for. But honestly, my inspiration comes from this Nature article. So in 2011, they stated that there's 8.7 million species of eukaryotic organisms on this planet. But the detail is really important. 80% of species still undiscovered. The enormity of eukaryotes on this planet have yet to be discovered. And I'll get to the details in just a little bit. But just consider that for a second. Everything that we know right now, this is 10 years old, but I'm pretty sure that number hasn't changed by an enormous amount. Everything that we know in terms of the living world, there have yet to be discovered an order of magnitude more. Okay, so we start with measurement. My first introduction into like scientific experimentation was actually partly out of laziness, but I wanted to grow my bacteria, but I didn't want to spend too much money on media. So I started going down this rabbit hole of making my own synthetic media, like Gatorade and Instant Ocean and things like that. So microbial growth curves, here's the theoretical curve, hangs out for a little bit, turns on the enzymes necessary to do metabolism. The cells are too low in count to actually count them well on most spectrometers, so you see this nice flat line. Then they start growing exponentially, where the cells are dividing rapidly. The slope of that can determine the actual growth rate. As the media starts as the food in the media starts to decrease, or in the quorum sensing within the microbe population, it starts to decline in population. And then once the food's completely out, it starts dying. Now, here's the theoretical model. Here's real world growth curves. So you see big fat error bars. There's a sample taken every 2 hours by some poor grad student who's forced to do this, or they just tag team for a whole day. We have better tools now, but this is basically how growth curves are presented. Growth curves are essentially cloudiness of media over time. So the cells divide, they occlude the media itself, they reflect light, they scatter the light, and the media gets cloudier, and therefore the amount of cells grow. Ish. It uses to measure this, we use a spectrometer that just basically shines light of a particular wavelength using a grating that has like a CD-ROM of sorts that reflects the light and allows only a particular wavelength to shine through, normally 600 nanometers for convention's sake. A little bit of math, nothing too crazy. But the main problem is that if you want to explore the natural world and you want to learn, okay, I have this microbe that I found on my shoe, what does it eat? Is it eating well enough? Is it growing well enough? How can I learn more about it? You need a device that costs around like $2,000. So out of my laziness, I made a $2.50 version of that, a tiny optical density unit, a turbidometer of sorts, with an LED, a light sensor, and a little bit of electronic smoothing. And we get a growth curve like this. So this is taken once a second for the entirety of its growth. This is a little bit over 24 hours in E. coli K12. But what was interesting when I first saw this plot, I was super excited because I'm like, wow, that's really low noise. But also, what's that blip? A little bit hard to see when it's blown out, but there's this tiny about 5 to 20 minute plateau in LB media. And I keep on seeing that over and over and over again. Similar to kind of a dioxic shift going on with Monod's PhD thesis in 1942. He was mixing different sugars, and he saw that certain sugars are preferred more than others, and the bacteria would deplete or get to a certain level of concentration of sugar A before it eats sugar B. Therefore, you get a plateau. So this could be some type of a dioxic shift. We're exploring it a little bit further just because we've seen these exact blips in published data before, but no one's addressed it. And also, it appears in complex media, not that much in defined media. So we're exploring that. Now, this was in 2015. Fast forward to a couple months ago, and my friend Philip Kyriakakis, at Stanford, he asked me to build this thing. You saw I'm really prolific on Twitter. I talk about all my research. I show that I do bespoke stuff. And he reached out and said, hey, can you build this? So this is called the Evolver machine from Boston University. It's a 16-channel directed evolution machine. It uses like I said, 16 channels, so 16 vials of media with bacteria in it, and this really expensive microfluidic plate to traffic all the fluids around. In parts, it's about $4,000, and I recently just saw the kit for $24,700. For many labs, this is breaking the bank, right? For PIs that are just starting, this may or may not be a large chunk of their startup. So I was tasked to build from parts. So this is an open source design. So we can order the PCBs and get all the parts and assemble it. But then a chip shortage happened. Here's one of my favorite optical chips, and it's back ordered until August 2023. And this keeps on getting pushed back. People keep on buying parts. So building this Evolver from parts, about $2,000 worth, was almost impossible. We could not find the parts. So I asked him something a little bit risky, and I said, would you let me design my own version of this, slightly more simplified version? So he said, how confident are you? I'm like 85%. He's like, okay, good odds. So I got an Arduino and I started cracking. I started thinking about simplifying this to its base components. What is this machine? So the Evolver machine involves a vial, a series of inputs of liquid, an output to remove waste, a magnetic stirring device, and a heater. And you multiply that by 16. Now I said, let me just simplify this and make a single one. So I used the PC fan, I glued some magnets onto it, I put a magnetic stir bar, and it started spinning at about 5 millimeters off the surface. So I'm like, okay, here's the start of something interesting. So fast forward a couple weeks, and I made this thing. So this was the little prototype of the Easy Evo. We called it the faux volver at the time, as just a little poke to the evolver folks. And it is, at first it was just a optical density machine that would just measure optical density of the culture over time. The evolver evolved, the faux volver evolved, and it became a little bit more complex, it became more modular, until what we have today. And so today I have 3 possible medias, 1 waste bottle, 4 motors, and it can do optical density with specific dosages down to the milliliter accuracy. So you can do interesting things, which I'll show with some fresh data that I got just not a couple days ago. So the first step was to do a growth curve. So here I tested with Vibrio nitrogens. It's one of the fastest doubling microorganisms on the planet. And so the faster the growth curves, the faster I can debug. So instead of using E. coli, I use this. So I let it grow overnight, and I got a nice clean curve. And then I activated the actual turbidostat module. So here is it growing to an optical density of 0.6, and then refreshing down to 0.3, and then oscillating. So this will keep it in a steady state between the two. And because the project is about optogenetics, we're going to be forcing a light switch to evolve using this type of activity. So we would allow it to grow in a particular medium that's selective, and then turn on a light and slowly redshift it towards our target wavelength. But in the actual test runs, I was really interested in salinity. So the first step, I let it cycle a little bit and then lag off, just like the previous one. Then I wanted to make sure that it's reproducible, so I did many cycles, and I saw that it was good enough. And good enough biology is fine by me, right? This is a prototype that I need to short the noise out. But this was in the same consistent media. Now again, there's 3 possible media combinations that you can add, and I decided to go with salinity in this case. So here is Vibrio nitrogens growing in 30 grams per liter salt, so LB media with 3% salt, sodium chloride. And then that little blip down there is when I shifted media over from 30 grams to 100 grams per liter. And you can actually see this data accordion stretch out as the cells are getting slower and slower in terms of growth. But notice that there's still an exponential phase. So we have this continuous log phase. And the more interesting thing is that as you crank it, as you allow it to adapt to that salinity, you actually see the accordion start to shrink again. So there's widening, a little bit hard to see, but this widening of the growth curve started to compress. And then I ran out of media over there, so I just kind of let it grow to make sure that it wasn't just this weird fluke of the machine. And then I decided to push it a little bit further. So here is already adapted cell culture at 100 grams per liter, and then push it to 120. As soon as that second dilution hit, it started doing something strange. There's this, the noise was due to clumping of the cells. So it's occluding as it's rotating, so it's causing these shadow effects. So the noise can be an indicator of cell adhesion, which is cool. But more importantly, what's that tiny plateau? And then the death curve over there is very particular, and the actual speed of that death curve, like why at this particular optical density and not another one? So there's some questions to be had. But the best part of it is that we took a $24,000 kit and turned it into a $163 device that you can 3D print. There's no custom PCBs. This is all commodity components you can buy off Amazon. So it's like directed evolution in the time of chip shortages. What can we do with that? So if you want 16 channels, build 16 of these robots. It's still cheaper than one of those. Now let's go to reading. So like I said before, the tree of life is way more complex. So we have eukaryotes down there, and we have this giant explosion of bacteria. And my favorite are over there in the candidate phylas. These are things that haven't really been classified yet. We don't even know where to put these things, right? And we're getting more and more of these as metagenomics becomes more popular. We're getting tons and tons of these candidates, these microbes that have never been seen before. That's kind of breaking our reality and where we place things. Like my favorite are archaea, because they're just so strange, and the extremophiles are wonderful, but people forget that we know of archaea as being extreme because those are the easiest ones to collect. What are the ones that are slipping through our fingers? What are the ones that can't be cultured on solid media? And so to go there, and in the theme of reading, after, let's say you do a directed evolution campaign and you're doing this in your bedroom and you say that something has evolved, well, you'd have to prove that. What if somewhere within that population were cells that are already capable of handling 100 grams per liter of sodium chloride? Sure. So here's the dawn of next generation sequencing. So I bought a nanopore, and it shipped residential because their motto is anyone, anytime, any place. So we got a nanopore, and we decided to try our hands at sequencing. So the story for our sequencing starts with this. So this is the Bombay duck, which is a fish, found in the Ganges River. And in 1962, a very special microbe was isolated called Dinococcus radiophyllus, not to be confused with radiodurans. Radiophyllus is way more radiotolerant, but nobody cares about it. So since the 60s, up until very recently, it's never been sequenced. It's been stored in the ATCC, people have done some research on it, but because all the funding went to Dinococcus radiodurans, people didn't really catch up with it. And conveniently enough, Carolina Biological sells it for about $12. And we noticed it hasn't been sequenced, so let's said, well, let's do it. It's a bacterial sequencing project. It'll be a weekend project. Thunder in the distance. It turns out that just isolating DNA took us about 6 months. So we used a super fancy $500 magnet bead kit, and it was garbage. We went through the optimization after optimization, and finally, after a tremendous amount of time, labor, and hours not slept, we got to the point where we got beautiful, ultra-high molecular weight DNA. The upper rung of that ladder is 45 kilobases. The smearing was okay. There wasn't anything issues with that. What you're seeing here is actually a gel visualizer that I hand built out of LEDs and some scrap plastic. And we got to sequencing. And so we did our first nanopore run, and we got our data, and they were like, okay, what do we do now? So we have this ton of data, like over 300 gigabytes at the time, and we're using a rapid kit that is about 4 generations removed from the current technology. So this was before nanopore even got a community large enough to answer these questions. We were in the blind. A lot of the tools there were in like version 0.1. And so we threw our bodies against the coals for about 2 years, and what resulted was this. So this is the full genome submission for Dinococcus radiophyllus, submitted by us, 2 degreeless heathens in a bedroom lab. And it was so good that it became the representative genome for that organism. So if you go on GenBank right now and you type in Dinococcus radiophyllus, our genome will be the representative for it. Not too bad for people who don't know what they're doing. So while we were on that high, we're like, okay, let's go to plants. Bad idea. So we found, I love this plant. So this is Oxalis Stricta. It's found everywhere. It's a cosmopolitan weed, meaning that everywhere where I've ever been, I've always found Oxalis. I ask my friends who live in different places, do you see the heart-shaped leaves? And they're like, yes. Its origins have been hotly debated, but the most important is that the identity of it as a species have been debated since the 1500s. Linnaeus thought it was a species. People argued for a really long time, but nobody actually wanted to sequence it. So after doing the bacterial genome weekend project that took 2 years, we decided let's not get too high for our britches and just sequence the chloroplast. So we isolated everything that looked like a chloroplast, assembled it, and I present to you today the first documentation of the Oxalis Stricta chloroplast genome. Ta-da! But more importantly, proof that from the 1500s until now, the people who are saying that it was a species it's true. We're going to have a preprint, a really nice small little blurb soon that shows the phylogeny of it and actual proof that it's a species, not a strange ecotype or isomorph of other Oxalis. And then once we did this, we had enough experience with nanopore that we decided to stare out the window. So here's the East River from my apartment. And this is one of the most wonderful places on Earth, even though it's a highway for shipping. It's actually because it's a highway for shipping. There's 8 million people. That means 8 million people are pooping on that island. And because it's a combined sewer outflow, that's going into the river. It's not called Flushing, New York for a reason. Not only that, but there are international freight lines that are coming through New York Harbor. The East River is not actually a river, it's a tidal estuary, where the Hudson River mixes with the Long Island Sound, the Atlantic. So you have these mixes of isohalines, these like salt density superhighways, where organisms from marine ecological niches and freshwater ecological niches mix, where the salinity changes often, where inflatable flamingos are shipped, just the strangest things you'll find in this place. And one of the most beautiful things about this is that nobody cares about this place. because it's garbage. It's where the mafia dumps the bodies. It's just this like random assortment of water that boats pass by. But we also have, New York City has the largest public school system in the world. We have trained researchers that are in top flight universities here. And a lot of people are doing wastewater management, wastewater screening for COVID. They're doing coliform levels to see what the toxicity levels of the water is. And because of the efforts of several people over the last couple decades, the East River has been the cleanest it's ever been, right? Since like we've started measuring water, since we started measuring any type of water toxicity. And so we have this giant pool of change, this untapped resource of organisms where you can start just taking a cup of water, and with careful consideration media, you can isolate, you can avoid growing the creepy brackish water human pathogens and focus maybe on the photosynthets, or focus on things that are extremely halotolerant or extremely sensitive to certain conditions. So you can make it selective enough that every single colony that you find there could be a new organism, and you could probably name it. Next topic, right. So we talked about reading, now we're talking about some of the projects that I use writing with. So here's a fun one. This is Rasica Chromoflora. So this project actually started out of spite, which is always a fun motivator. So real quick, what color is this? What color is this rose? Purple. Cool. Would anyone say that this is blue by any chance? No? Okay, so would you believe me if this is the Applause Blue Rose, the first transgenic blue rose by the Suntory Whiskey Company in Japan? So they call this blue. This frustrates me to no end, because in kindergarten I was taught what colors are, and we have a mutual understanding of color. And I get it, it's like a cool lavender at best. I promise I didn't screw with the labels, just type in Applause Blue Rose. And it got a lot of publicity, and they sell for like $25 a stem. I'm like, that's not blue, these are blue. So here's a tropical clam, the Maxima clam, where the lips have these beta barrel proteins that line up really well with GFP. In the center is green fluorescent protein and a bunch of other blue chromoproteins, and you can see them lined up. I ran them through RaptorX to do protein alignment, and they look pretty close to GFP. And that's great, because we've been expressing GFP in every living thing we could find. It's like the cool thing to do. Now, on the top left is the Gus assay, a really common histological assay for plants to show expression of promoter expression. There's an enzyme that turns a colorless liquid blue into a blue precipitate, similar to the technology found in cyanotypes, old photography technique. On the bottom right is a computer visualization of what a blue rose might be. That's not that interesting. The top right is interesting, because that's a blue strawberry. But because it's blue, it's not really that strawberry yet. So we can call it whatever we want. So I was giving a lecture to some second graders as a scientist in residence of sort for about a week. And I talked to them about this blue thing. And I asked them, okay, if we make a blue strawberry, what should it taste like? And we had a vote, and there was, it was a tie between bacon and bubblegum. So I don't know how I'm gonna do that, but I have like the most discerning clients in the world now that are going to judge the crap out of me if I can't deliver on a blue strawberry that tastes like bacon and bubblegum. And we can call it snozberries. We can live the dream, why not? So in the age of synthetic biology, I'm really upset that we don't see examples of really crazy looking flowers. A lot of them are discarded because it was just a weird mutant, but I wish people would collect that and just have this library of mutants, a garden of GMOs, properly contained, following USDA guidelines, but still. So in my quest to make blue, I found some blue chromoprotein sequences. On the left is the wild type expressed after a couple point mutations and some series of like brute force directed evolution, plate to plate. We got this nice dark thing. And today I have this. So what color is that? Blue? Yeah, cool. Okay. So not purple, right? Cool. But I fell in love with chromoproteins so deeply because there's so much potential without the need for fluorescence. Like what if we do pink, right? This is MRFP1, the monomer of DS red, visible in daylight. I started playing with yellows. Number 14 looks beautiful. That's my favorite. So this is my yellow. So now I have a blue, let's call it a cyan, a yellow, and a magenta. There's something missing, a black. So it turns out that in nature, black's really hard to make. But melanin gets really close. So there's a microbe called Rhizobium etli that has the MEL-A gene, And it is a tyrosinase. It chains tyrosines together. The more they're chained, the darker the pigment gets. As it expresses, it occludes more light, and it gives you the impression of black. And I said, okay, science should be reproducible, right? So I found the sequence, I ordered it through Twist, and I tried it in bacteria, and ta-da, it works. So this expresses over the course of a couple days, so it still needs some optimization, but you now have a cyan, a magenta, a yellow, and a black. That's the CMYK color spectrum that we use for printing. That's the subtractive color space. So now with these, essentially these 4 genes, you can now start mixing them together in terms of expression and get all the different colors in between, just like how printers use just 4 cartridges. So the first thing I did was I did a crude extract and I mixed them by hand. So here's those 3 proteins mixed in 20 microliter increments. And you can see really cool colors, like there's oranges and peaches and reds and that green. I know plants are green, but hear me out. Imagine fusing the blue and the yellow together to get a green chromoprotein that's not metal binding. You don't have to give it copper or magnesium or whatever. It just is green because you have those 2 and they're right next to each other. So I said, you know what? What if we put it all on a plasmid, a single plasmid for plant transformation, and have students make synthetic promoters that modulate the amount of expression of each one of these promoters such that you can get that Rasica Chromoflora, so that you can actually get those colors, all the colors that you'd want with just 4 genes. Instead of diving through the delphinidin pathway to get blue, why not just express these proteins? So I found the Wisconsin fast plants, which I love. They're 15 days seed to flower, 30 days seed to seed. It's been an established part of educational curricula for 30 years, but it's never really been pushed to biotechnology. So I contacted the folks at Wisconsin-Madison and got in touch with them, and I'm actually slowly pushing that idea of first sequencing this thing because it's heterogeneous for everything. It's an absolute mess. But one of the grad students there told me that they have a line that's nice and clean we can start with. Develop the tissue culture protocol for it, and then start working with educators to see how we can incorporate this into curricula, how we can get people to transform plants on top of just transforming poop bacteria that turns beige to green. Why not have flowers that produce different pigments? What if you can spatiotemporally localize those pigments on a petal? You can start designing here. Along with those colors, I've made some in-betweens. So here's orange that I took from some wild types with some point mutations. There's some ones that are already published. All of these are open source. Shameless plug, I sell them. DM me, I'll be more than happy to sell you these. They're constitutive and entirely open source with no MTA, 20 bucks each. Okay. So that is the pretty stuff. Now on to something a little bit more serious, a lot more serious. So antibiotics. So this is a problem, obviously. Feedlots are one of the largest contributors to the antibiotic crisis, misuse and overprescription of antibiotics in medical settings, sure. But regardless of this, this is still happening, right? So we have bacteria that used to be resistant. All these discs are different antibiotics dried on discs. And you see that they're getting more and more acquainted to their media. They don't care about those antibiotics as much. Bacteria are super clever. They have specific pores. They have specific pumps that are dedicated just to certain antibiotics. They have all kinds of different defense mechanisms in which they can adapt or break down, like beta-lactamases. They can break down most silins. And it's getting worse and worse and kind of scary. Now, as many of you know, in molecular biology, we use antibiotics a lot, specifically in selection. A lot of our plasmids that carry our genetic payload also have an antibiotic component attached to it. Ampicillin, kanamycin, vancomycin if you're nuts. And we use it in liquid media too, where this effect gets even worse because once this is done, if you're in an amateur setting and you forget to autoclave it, that goes down the drain. You're adding more and more possibilities for the microbes in your drain, for the microbes that you're putting in there to enter our ecosystem one way or another. Now, the amount that academia produces in terms of antibiotics doesn't really go into the ecosystem because you have incinerators. You just put all your stuff in the biohazard bin and it goes away. We don't have the away. So what amateurs like myself have to do, and I hope they're doing it, is to autoclave the crap out of the media and make sure that it doesn't work before getting rid of it. But I came up with a silly idea that I think will work. Sugar. So it turns out, weirdly enough, almost all of the common laboratory E. coli strains lack the CSC operon, the sucrose metabolism operon. They can't eat sucrose. So I said, why don't we take the CSCA gene that is a sucrase that basically breaks sugar into glucose and fructose, and express it in a plasmid and see if it works. So I made minimal media where the only source of carbon is sucrose. I did some tests prior to with my strain. It's NAB turbos. I did E. coli K12, DH5 alpha, top 10, 10 beta, a bunch of other strains. I also found their genomes published, so I made sure that that gene isn't actually there, and it worked. So to the left has the CSC gene, to the right doesn't. The media has only sucrose. I also added a pH indicator because the metabolism of sucrose actually breaks down. As sucrose is breaking down through the metabolic process, a proton is donated into the media, acidifying the media. So the media is containing chlorophenol red, which goes from a pink to a yellow if it's also acidifying. And those colonies are pretty yellow. So I said, okay, why don't we do what regulators might like and remove the antibiotic gene entirely and make a plasmid where the sole selection agent is the presence or absence of the CSEA gene. And I made sure that when I synthesized this gene, I ordered it codon optimized so that it doesn't actually line up with anything on any E. coli genome. That took a little bit of time, but this is already in production, and I'm working on making this available to people because antibiotics in the United States you can buy for aquariums. You can get kanamycin, pure kanamycin that you can dump into your aquarium to solve like fin rot. You're diluting it, and all the microbes in that aquarium are slowly going to be resistant to kanamycin. Huge problem, no one's really talking about it, kind of bothering me. But in other countries, these are still frontline antibiotics, and it's incredibly difficult in other countries to get your hands on antibiotics. So if you want to be an amateur biologist, or if you're an educator and you want to get students involved in biotechnology, you would either have to go through tremendous hurdles to get your antibiotics, or you cannot have your students participate in biotechnology. And I don't think that's right. So a tool like this could be beneficial. What if you have this media component, a specific cell line, and a specific plasmid that is selected with sugar? And you can, with the wonderful world of synthetic biology, you can delete certain genes and make it not able to eat other sugars. And then now you have a bevy of selection agents that don't crosstalk. Now moving on to plants, you can do the same type of selection in plants. So in eukaryotes, the PMI, the phosphomannose isomerase gene, takes mannose that gets imported, that's mannose 6-phosphate, and breaks it down and turns it into fructose 6-phosphate, which then feeds into the Krebs beautifully. What's really cool is that many plants lack the ability to eat mannose. So Syngenta, many years ago, produced a patent for the PMI gene, the phosphomannose isomerase selection system, which is now off patent, which is great. The wonderful thing about the PMI system is that just like the sucrose selection, you now have the mannose selection, and with mannose selection comes acidification. So you can have all of your plants on a well plate. You can have little leaf punches of each one, put them on that plate, and overnight the media will acidify if it's positive. And the degree of acidification is proportional to the amount of expression of the phosphomannose isomerase. So a wonderful assay that people aren't using that often, and that I highly recommend. Now the third most wonderful thing ever is Ruby. So I don't know if many of you have heard of it, but it's a wonderful new marker that is purportedly non-invasive. It doesn't add to the metabolic burden, and I'll explain in a sec. So most plant transformation technologies start with this. So this is a leaf disc of tobacco. We expose it to Agrobacterium. This is a bacteria that normally causes tumors on trees. Scientist in the 80s, 79, with Mary Del Clinton, she produced the first proof of Agrobacterium as a vector for transforming plants. Fast forward to today, it's used everywhere with all kinds of different transformation technologies. Tobacco is one of the easiest, it's the easiest to learn. And the process is like this. You soak it for 20 minutes in Agrobacterium that's made angry. They act like sharks in the water, but if you use acetylsyringone, which is a very expensive molecule, you can activate the virulence pathway, or you can look for acetylsyringone homologs. One of them is vanillin, so vanilla. So I took some vanilla from my mom's pantry, got them really angry. The smell of the culture changes when they're angry, so you can tell, and they don't smell like vanilla. You plate them on a culture, and you let them sit for about 2 days in the dark. And now these halos start forming as the bacteria swarms around it and starts growing. The media contains sugar and nutrients for all life to grow. This is MS media, a really common plant media. And then you move it onto a plate containing herbicides. In my case, it would be mannose, because I'm transferring the PMI gene. But one of the biggest problems is that even if they're growing, how do you know that these plants are transgenic? How are you sure? You'd have to wait until they grow a little bit, PCR them, and pray that there's no residual agrobacterium on your leaves, because that'll give you a false positive. So it takes a while to actually figure out whether or not the gene was integrated. And if it's positive, enter beets. So beets are red because of betalains. So most plants either have anthocyanins or betalains as their purple pigment. There's never been a report that there's a plant that has both, so it's a nice segregating gene to pay attention to when it comes to figuring out what the hell this plant is. Now, in 2020, a man by the name of Yunde Zhao in his lab produced this wonderful circuit called Ruby, a single transcript, 3 enzymes that converts tyrosine, our friend from the melanin circuit, and converts it to betalain, adding a sugar at the end. So these 3 genes, when expressed in any organism that has tyrosine in it, and tyrosine is produced in the chloroplast, you get this. So this is the first cell I've ever made with the Ruby construct. This is about maybe 13, 14 tobacco cells. And if you culture them, they start growing. And what's really nice about Ruby is that it's a very, very, very stark difference between wild type and transgenic, red versus green, especially in tobacco. So without even any herbicides, you can just start selecting the red tissue. And the tissue starts growing and growing. Some of them do the strangest things. I love this transgene so much. There's selective silencing. If it hits the meristem just right, you get this type of split phenotype that I'm really trying to stabilize. I would love to see that. This is under the 35S promoter, so it's not tissue specific. It expresses in all tissues. Its roots are ridiculous. Like, they're the most red, beautiful things. This isn't a modified image. They're very, very healthy. There's not much in terms of invasiveness of the reporter. It's not stunting growth. And the most gorgeous bronze plants as a result. What I was really worried about tobacco is that obviously it's green because it has chlorophyll, and I thought the chlorophyll would compete with these colors. But look at that stem. That's red. I don't see any shade of green on that stem. On the leaves, sure, but the stems are typically green. Here's an example of a strong producing phenotype versus a weak producing phenotype. This is all from the same experiment. I did it once. The flowers are also entirely pink, as opposed to those tiny little surface pinks of the tobacco. And these coupling together produce the sucrose selection in bacteria, the sucrose selection in agrobacterium, if we knock out the CFC gene in agrobacterium, the phosphomanose isomerase in plants, and then the Ruby marker to visually indicate, without needing to use herbicides, what tissue is transgenic. Couple them together, and you have the beginnings of a sandbox. A sandbox that I would love children to explore, high school students, maybe even middle school students with good guidance, to be able to one day produce something like this, right? So this is from Luigi Serafini's codex that Maya Hay talked about. This looks ridiculous at first, but if you just consider for a second that outside of the one that with the cloud that's raining on itself, that's silly. Not all of this is outside of the wheelhouse of what nature can do. And this is something that synthetic biology can utilize. This is something that with synthetic biology we can achieve as well. Not just for aesthetics, but imagine seeing the look on a high school student's face when they produce a flower that no one has ever seen before with their own hands through this open-ended sandbox of play. Now the last part, really important, sharing. So we did measurement, reading, writing, and now sharing. As an amateur biologist, I don't have the funds to publish in formal journals, but we have wonderful alternatives. BioRxiv is the greatest thing ever invented. It's free, you upload it, you get comments immediately. Science Twitter is one of the greatest experiences, intellectual experiences I've ever had. I thought it was just gonna be like a bunch of professors yelling at each other. It's the most warmest and inviting environment when it comes to research culture. It's the reason why I'm here. The bulk of my income comes from contract research through contacts on Twitter. We also have Zenodo and Figshare. If you just have a little clump of data that you want to publish, you can just slap that into Zenodo and you get a DOI number, digital object identifier, which gets permanently archived forever. If you die, your work is still there, instead of it just being on a blog somewhere. I highly recommend you publish. It doesn't have to be with a journal. Also, when it comes to code, GitHub, obviously, one of the greatest repositories of this. And so I I hope that with small, thoughtful science, more and more amateur biologists can explore all of the ungrantable questions that academia may not touch on, all of the wonderful things that we don't know. So thank you so much. I'm a tiny green cell on everything, so feel free to follow me. Use my email. We're going to also host our genome data on Pirate Bay, which will be cool. Yeah, download our genomes on NCBI, follow me on Instagram, Forcus on GitHub, and just say hi. Thank you so much.
Go for it. Yeah. It was the best talk ever. It was so good. So more like comments.
Sure.
I love your CMYK color palette. It's very tidy looking. And you know what I'm talking about, right?
Yeah.
Okay, so that was great. And you do a phenomenal communication job on Twitter. So you guys follow him on Twitter, Tiny Green Cell, and the threads that you make, and that's my go-to source for a lot of science, new papers that are coming out and all of that. So those are just my comments.
Thank you so much. I appreciate it. Good work.
This was really like mind-blowing. I really liked everything you showed, and you're just really inspiring as an individual in general. But I had a question with your project, with the collaboration with the University of Wisconsin, the plants that you wanted. What do you expect to see with that, or what are you trying to actually do? Like, are you trying to get plants with all the different colors on them? Like,
okay,
or
that's a good question. So, when I introduced the Wisconsin Fast Plants, I said that it's seed to flower in 15 days. So it's a rapid prototyping tool for flower modifications. And not just pigments. The pigments are the first start. There's the SAT42 transcription factor, where if you mess with it, you get crazy morphologies. I'm more interested in the morphology. I'm just like this big, and I need to be this big to learn about plant morphologies effectively, right? So we're starting with baby steps. And what I'd like to do is to piggyback off of the already established educational curriculum around genetics and Wisconsin Fast Plants, which, like Carolina Biologicals, distributes curricula. There are many, many universities, especially CURE curriculum participants, that use Wisconsin Fast Plants to track allelic variation, to track heredity, to teach basically advanced Mendel. And so why not utilize something that's tried and true that educators have an experience with and add one more component? And there's a wonderful professor, Margaret Young, from Elizabeth City University. And she was the first to show Wisconsin Fast Plants regeneration from a particular tissue type that isn't immature embryos. So she's like pioneering the ability to modify this. And I want to collaborate with her to be able to introduce this into a high school curriculum or early undergraduate curriculum, where you go from, I grew these plants when I was a kid, to I've now made a flower no one's seen before using that same model. So little kid, little question, big kid, big question.
that's really cool. Yeah.
So you were talking about including a lot of these things in a high school curriculum. Well, you have a high schooler right here. So my question is to you, what kind of call to action would you recommend for a high schooler looking to get into molecular biology, whether it be human or plant? And how would you recommend for someone like me to explore this field when it's not usually taught in class?
Okay, that would be a nice and long discussion. And I would love to have that conversation with you, because there's nuance depending on what high school you're in, what geographic location in the world you are. What I'm doing in my home lab would be a federal felony in Germany, as an example. So yeah, so depending on where you are, the legalities are drastically different, right? In the United States, biosafety level 1 is an interesting legal framework. There's not much of a penal code. So as long as you stay within biosafety level 1, there's plenty of things to explore. And the first thing I would ask is to ask your educator if there's room in their curricula for things. Because one of the issues with high school is that you have to learn so many things and then apply for college, right? And there's this mill where there's very little room for these type of extracurriculars, right? One of the main reasons why I started doing research at home is because my high school was dead broke. We didn't have any time for any type of a lab component, and I was still very curious, and I learned through hands-on. And so if somebody tells me this is what a cell looks like and you have pretty diagrams, if you don't see it yourself, it doesn't really land right. So I would talk to your educator and say, hey, I'm curious in this, can you point me to the right place? If not, there's this wonderful concept called community labs. They're popping up all over the world, right? Where there are biohacker spaces guided by folks that are established researchers, maybe retired researchers, that can help to make a sort of gym membership for biology, right? It's like you pay a monthly subscription and you have access to the lab, and you can hop onto a project already in progress to kind of sharpen your tools. If you do decide that you want to build a home lab, I strongly recommend you build it around a question. Don't be a generalist. I was burned terribly by that, right? I just became like an equipment fetishist. I'm like, oh, I have to have the nicest equipment. And that was a silly concept. Start with a question, because the first part of any biological project starts in the library, not in the field. So come up with your question, build around that, find what equipment you might need, reach out to professors, reach out to them on Twitter. They're actually really nice, and say, hey, I have this idea. What can I do? Can you point me to some papers? Because if education doesn't give you the mentorship, you can reach out to people who are passionate, or people who they see themselves in you. They see this passionate person they used to be before they got ground down by the grant mill. You know? Not to disparage any academics in the room. Yeah, so do that, but also contact me on Twitter and I'll let you know more stuff, for sure.
Thank you.
No problem.
I'm wondering if I can jump in and kind of piggyback on that question. First, great talk. I'm thinking about the biosecurity type conversations that are happening, you know, within folks like GP Wright, and who are really wary of precisely these type of biohacker spaces and are perhaps unnecessarily vilifying them. And I'm thinking also specifically of like people like Josiah Zahner. And I'm wondering if you can comment what your experience has been in terms of that type of regulatory framework, and even just like unofficial regulatory stuff, right? That sort of in-group policing around like who is available and who is not allowed to do this type of work. I see that, you know, you've tapped into Twist and other folks. And so, yeah, I'm wondering if you could speak to your experiences. I'd be nice to hear. And yeah, great talk. Thanks.
Thank you. Okay, so where to begin with that? So first off, I've been a part of the DIY Bio Forum since like 2008, right? And I've seen ideas come and go, radical ideas, not so radical ideas. And what I've noticed is that talk is cheap and execution is difficult. And more often than not, we want to create rather than destroy as a community. And I've seen so many examples of people that want to make beautiful things, that they want to feel inspired about the natural world. They don't want to build a virus. They don't want to hurt themselves. Ironically enough, some of the most well-trained and some of the most well-trained members of the DIY biohacker space tend to be the ones who are the most provocateurs, which is strange. So it goes to show that having a PhD doesn't necessarily imply a sense of ethics. It kind of sometimes is decoupled. But when it comes to regulations, like for example, the FBI started an outreach program for DIY Bio, a voluntary outreach. And so, hi Sarah, if you're listening. I have a FBI agent in New York that I meet like every 6 months or a year. I haven't seen her for a while, I should. And I basically say like what I'm doing. She buys me a burrito, it's nice. And more importantly, she wants to know like if any shady, strange characters have approached me. She's not worried about what I do. I'm an open book, and most of the people who are doing biology love to share this, and they are open books too. And when somebody says, oh, I want to do something in a mouse model, they go nuts. Everybody's just like, that's a terrible idea. You shouldn't do that. You don't have the equipment for it. You don't have the experience for it. No one's going to oversee what you're doing. So there's a lot of self-regulation that comes with this community, because it just goes to show that human beings are curious, and only a lower percentile of people are actually wanting to do bad. It's also very, very difficult to do anything with biology. So doing something bad requires this like elegant evil that's just, it doesn't seem practical. When people say like, oh, I want to make a glowing plant, that more people are interested in glowing plants than making some type of a bioweapon by orders of magnitude. And that one dude who said something about bioweapons was excommunicated from our group immediately. Yeah, so there's a ton of self-regulation, and when it comes to vilification, this is my like call to action for other amateur biologists, is to showcase the good that you're doing, because you can't have vilification if the evidence is to the contrary, right? I'm not on the stage to like flex and be like, look how smart I am, I'm doing this without, no. The idea is that anybody can do this, anyone can contribute meaningfully to the scientific conversation, and the bulk of what the contributions people are talking about are things for the greater good, or for entertainment in a way that's actually safe. So yeah, in conclusion, this kind of a roundabout way of answering her question. But the key point is that there are just so many things that one can do with all this power at their disposal, that doing something evil would be just so boring.
Agreed. Well done. And also, yay Arduino and Raspberry Pi.
Yeah. So I'll say, take the Q&A question first,
Sure.
if you can.
I don't have control of it. Okay. Very specific question here. Could you talk a little bit about the RDD displaying viruses? I don't think this was for me. Oh, this is you? Okay. Yeah. I don't know enough about viruses. Yeah, I'm a plant guy, I'm sorry.
And do we have anything here? That's Maya's comment.
Okay, cool.
So I will run and go the other way to disparage the academics. What are some systems that academia puts in place that stifle your progress, and what ways could academia adjust in order to help?
Sure. So, like I said, I'm a degreeless heathen. I don't have a degree. That infernal parchment has blocked my career progress in science more than anything ever. Getting into a graduate program without a bachelor's is damn near impossible. All the exceptions that professors, good, well-intentioned professors offer me, flop in front of the HR department that says, "Thou shalt not go to grad school without a degree." Regardless of publications, track record, experience in industry, it has been impossible to do anything in terms of advancing my education. So I decided to just advance my abilities as a biologist and collaborate with the very people that I would have been learning under. So it's an interesting roundabout way where I became my own mentor, which is unfortunate, because the only thing that I really crave that's truly missing from this experience is mentorship, right? What was that old saying? The product of a PhD is not a dissertation, it's you. It's how you are shaped as a better scientist in an ideal world. And so that type of experience is sorely lacking. So my Twitter family is my de facto defense committee. I started this impromptu PhD thesis on the sugar selection end-to-end, plant genetic engineering using without herbicides. And they've been humoring me with mini little mock defenses and stuff, and one day I'll live stream a defense, and at the end, publish a tiny little book about everything that I'm doing. Again, not for the accolade. I don't need an accolade, I need mentorship. So this lack of the ability to go to grad school blocked me from getting that mentorship. And so what I'm trying to do to mitigate that, not for myself, but for the self I used to be, for the amateur biologist who didn't throw their bodies against the coals sillily for 15 years, is to get a group of established researchers to evaluate manuscripts that may or may not have been written by a 15-year-old, a 12-year-old, a retired plumber, right? A kind of pre-print prep. Just like how an English teacher circles with red pen, saying, okay, this part's crap, this part's crap. Give somebody a way to apply rigor to their curiosity. Please.
Actually, I do have a few comments. And gosh, for the very first time in my life, I do regret that I don't have a Twitter account. I'm such a private person. So, and another comment, you mentioned a couple of times, twice, that you are lazy. Come on, I disagree. So you have a very sharp, bright, a very quick mind, and incredibly open mind. My third comment, just please establish more connections with academia.
Yeah. No.
Don't quit.
No, not at all. No, regardless of what my station is in life, I'm a biologist and I want to continue doing research.
Yeah, I don't envy your mom, and I can imagine how
she's shout outs to my mom. She's the greatest lab manager.
Okay, so ever. I say hi to her actually, so my respect to her.
Yeah, no, she's my greenhouse manager, my lab manager. Okay. Yeah, it's incredible.
It's good that you do share your passion. So, and then speaking about the colors, so did you think that, it could be applied to produce more gene reporters? We're so much interested in that as a basic tool in the molecular biology field.
Yeah.
So just go ahead and
every single marker that we'll be making for flowers will be a visual marker for plants. Yeah.
And then not only for plants, for molecular biology. So we can use it as the reporter gene.
Yeah, and because it's open source, people are free to do directed evolution, make it better, and kind of build this ecosystem that for some reason we don't have yet.
So excellent talk. Thank you.
Thank you so much. Anyone else? Cool. no problem. Thank you so much.
So with that, we want to, the Genome Writers Guild wants to thank everyone for coming here today, either virtually or in person. It's been great to talk to everyone and have a great discussion. I'm really happy for this public session going well, and hopefully everyone was able to learn a little bit and see something new from biology. So thank you everyone. For conference attendees, we'll start again tomorrow at 9:30.